Now, let's take a look at the cell component. This cell alone gives you a good description of what a eukaryotic cell is, and specifically an animal cell. This is what we call a generalized model of the animal cell that gives you all the structures that you can find in an animal cell. Now, I invite you to spend some time on this slide and to actually not only identify different components that make up the cell, but also to see how the cell is organized. You notice that the cell is a network of flattened sacs. That network of flattened sacs exists because there is a relationship between the different organelles. Some reactions start in one organelle, then they're going to move to the next organelle, and then to the next organelle until the process is completed. But I'm going to spare you some details here. Now, the organelles are defined as structures in the cell that perform a very specific function that another structure is not performing, right? These organelles are essential because they allow eukaryotic cells to separate different chemicals reactions to optimize the functioning of the cell. So the biochemical reactions are going to occur in different spaces to optimize the way the cell works. So some organelles are going to be specialized in containing the genetic information. That is, for instance, the case of the nucleus, right? And some are going to be specialized in producing energy, such as mitochondria. Some are going to be specialized in carrying the chemicals that are synthesized in the cell outside of the cell, right? This is the role of the network of flattened sacs in the cell. And you have some organelles that are responsible in breaking down the debris, the chemicals, when the cell needs to break down these different components. Now, first up, let's take a look at the nucleus. The nucleus, it's a structure that is relatively complex with a nuclear envelope. That nuclear envelope has some little holes that we call nuclear pores. Okay? When you look at the nuclear envelope, you notice that you have two membranes. So you have one membrane right here and another membrane right here. Now, what we notice, if we look carefully, is that the nuclear envelope is actually folded in this way. All right? So this is a phospholipid bilayer. This is another phospholipid bilayer, right? This is a double membrane, right? That is folded and with some proteins embedded in here, making up the nuclear pore. And that nuclear pore, it's the structure through which some chemicals are going to go from the nucleus toward the cytoplasm, and eventually from the cytoplasm toward the nucleus. Think, for instance, of enzymes that are responsible for copying the genetic information. The genetic information occurs in the nucleus, right? So all the structure that you see here, that's the genetic information, okay? So some chemicals need to get inside of the nucleus, others need to get outside of the nucleus. And that movement is made by way of nuclear pores. All right? Now, there is one particular area of the nucleus that is very dense. Right here, this is called the nucleolus. The nucleolus is the area of the nucleus where the ribosomal RNAs are produced. Okay. Now, in addition to that nucleus, that nucleolus, you're going to have the nucleoplasm, which is basically the equivalent of the cytoplasm, but this time inside of the nucleus. You're going to have some chromosomes, and chromosomes are going to be a mix of DNA and some proteins, right? Proteins that we called histones in the first chapter. And in the nucleus, we're also going to have some RNA ribonucleic acids. Now, the endomembrane system is that network of flattened sacs. That network of flattened sacs allows the chemicals to go from one compartment to another compartment to another compartment and separate different chemical reactions in what appears to be a process, right? So this is a series of membranes, right, that are interconnected. So we're going to have the nuclear envelope that is going to connect with the endoplasmic reticulum. That endoplasmic reticulum itself is going to connect with the Golgi apparatus, which is going to connect with the lysosomes and some vesicles that allow transportation of chemicals here and there. Now, the endomembrane system is a network, as I mentioned. So you have the nucleus here, and notice that some chemicals are being produced, going from the nucleus. We have some chemicals that are synthesized here, here, here. So you have a complex network. Now, in that network, some proteins are produced through what we call the endoplasmic reticulum. That endoplasmic reticulum that is involved in proteins production is called the rough endoplasmic reticulum. I want you to remember that. That rough endoplasmic reticulum is started with some ribosomes in yellow here. These ribosomes are the ones that are responsible for the synthesis of proteins. Once these proteins are synthesized, they move within the lumen of the rough endoplasmic reticulum to allow these proteins to be modified. When these proteins are modified, eventually they're going to end up in a transport vesicle. That transport vesicle is going to migrate toward the Golgi apparatus, where the proteins are going to receive further modifications. These modifications are going to allow the proteins to be properly addressed, whether they go outside of the cell in the process of secretion, or they go in the lysosomes in this compartment where digestive enzymes are going to be found and are going to actually allow the breakdown of all the chemicals and particles that needs to be discarded. Now, I mentioned the rough endoplasmic reticulum, right? Now let me tell you a little bit about the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is essential in the synthesis of new lipids. That synthesis of new lipids is essential for the renewal of the membranes, right? Okay, so the rough endoplasmic reticulum works in close collaboration with the Golgi apparatus, right? That is actually sorting proteins to send them where they belong. Some proteins are integral part of the plasma membrane or any other membrane in the cell. Some proteins are designed to go outside of the cell by way of secretion. Some proteins are designed to end up in the lysosomes as digestive enzymes. Okay? So please review the role of the smooth endoplasmic reticulum, the role of transport vesicle, The role of the Golgi apparatus, okay, the secretory vesicle, the lysosomes, please review all of that. Now, the rough endoplasmic reticulum, I already mentioned that it is studded with ribosomes, which allow them to be involved in protein synthesis, and these proteins that are synthesized through the rough endoplasmic reticulum get modified, right? The smooth endoplasmic reticulum, on the other hand, is moved because it does not have ribosomes attached to the surface. That's one, which means that they're not going to be involved in protein synthesis. However, they are going to be involved in the metabolism of lipids, the synthesis of lipid, the carbohydrate metabolism. And then I also mentioned the Golgi apparatus, which actually is important in sorting proteins, right? And if the Golgi apparatus has a problem, the proteins may not end up where they need to end up. It's like basically putting a wrong address on a package, right? But that's another story that I'm not going to dive in. Now, this slide here is to kind of show you how the network of flattened sacs in the cell function together for a major outcome. Now, here we are going to address the production of milk in a woman after giving birth. Now, that production of milk is going to involve a number of components. Now, from the nucleus, we're going to have DNA that carries genes. These genes are responsible for the many proteins that are found in the milk. You're also going to have lipids in the milk. Now, these genes, when expressed, they are going to end up forming proteins. These proteins are synthesized through the rough endoplasmic reticulum. They're going to be modified in the rough endoplasmic reticulum. And eventually, these proteins are going to be carried toward the outside of the cell. So here we have the rough endoplasmic reticulum. From the rough endoplasmic reticulum, we have here the Golgi apparatus, right? And so, again, the protein that are synthesized through the rough endoplasmic reticulum here are going to migrate in the Golgi apparatus. And from the Golgi apparatus, some transport vesicles are going to form so that these proteins can migrate toward being expelled outside of the cell. So that is the protein segment. Now, how about the lipid segment? Now, the smooth endoplasmic reticulums right here are going to end up forming vesicles like this. These vesicles are going to contain lipid droplets. These lipid droplets are going to come together in an ocean of water-based environment and are going to be sent out in this form so that when a mom is releasing milk, that milk is a mix of lipids, proteins, and carbohydrates, right? So I forgot to mention the production of carbohydrates here. So milk, it's basically proteins, lipids, and sugars or carbohydrates. So this is how the network of flattened sacs is working together for the production of milk. Please review the slide. The lysosomes, which is part of the network of flattened sacs, is going to be an organelle. Lysosomes are organelles that are actually going to contain digestive enzyme. This enzyme break down about anything. They break down proteins, they break down lipids, they break down carbohydrates, they break down anything you can imagine. Nucleic acids, they break down anything. Think, for instance, of a cell in your body that is responsible of engulfing a harmful bacterium or a harmful virus. When they engulf these particles, they're going to cut these particles into pieces, right? Now, they are able to cut these particles into pieces thanks to all these digestive enzymes, okay? And you know that a harmful bacterium, it's a bunch of proteins, carbohydrates, sugars, nucleic acids. All of that has to be chopped up into small pieces. Okay? And the digestive enzymes are responsible for that. Okay? Now, also, when some of our particles in our cells get damaged, for instance, a mitochondrion that is not good anymore, what becomes of that mitochondrion in our cells? Well, that mitochondrion has to be broken down, right? The lysosomes and the digestive enzymes in the lysosomes are the one responsible for breaking down those damaged organelles in a process of autophagy, right? The vesicles are going to be essential to carry the cargo from one place to another. So here we have the lysosomes, and as you can see, for instance, we have some organelles here that are damaged. The mitochondrion here that is damaged. These organelles that are damaged are going to be engulfed in some kind of vesicle, and that vesicle is going to merge with a lysosome, and the lysosome contain digestive enzymes, right? Contain lysosomal enzyme or digestive enzymes. Say we are importing some foreign particles, a bacterium, a virus. These foreign particles that end up in a vesicle, that vesicle is going to merge with this smaller vesicle that contain digestive enzymes, so that what you have what we call here phagolysosome. So let me spell it for you. Phagolysosome. Okay? This phagolysosome is going to be responsible for breaking down the foreign particle that come in our system and ready to be broken down. All right, so I already told you about the lysosome. Now, what happens when the lysosomes do not do properly their job? We know of some diseases in our system, genetic diseases that are caused because of lysosomes that do not carry the proper digestive enzyme. Such a disease is, for instance, the Tay-Sachs disease. The Tay-Sachs disease, it's a genetic disease that is that can be passed down to offspring, and that causes an accumulation of lipids in nerve cells, and specifically the nerve cells that are found in the brain and those that are found in the spinal cord. These nerve cells that become dysfunctional because of the accumulation of all these lipids are going to cause the body to not respond properly anymore. So in general, people with Tay-Sachs disease are going to die within the first 5 years of their life, okay? Because as these lipids accumulate, the nervous tissue degenerates. There is no coordination in the functioning of the body, and the individual end up dying because of that. One organelle I have not mentioned so far is the peroxisomes. The peroxisomes are those organelles that are membrane-bound organelles. They're going to contain many digestive enzymes. However, this digestive enzyme is not as numerous as the digestive enzyme you find in the lysosomes. That's one. Two, they are going to be very limited in terms of the spectrum of their activities. Most of them are going to be geared toward breaking down lipids, and some of them are going to be geared toward synthesizing the bile acids that are essential for the formation of bile salts. And the bile salts are necessary for the digestion of lipids during digestion, right? The peroxisms are also essential for detoxification. So most of the time, these peroxisms are going to break down alcohol, right? So that the toxicity of alcohol doesn't pose a problem to you. Now, the peroxisms are very abundant in the liver, right? To break down, for instance, alcohol. They are also very present in the kidney. One condition that we see as dysfunctioning of the peroxisms is the adrenoleukodystrophy. The adrenoleukodystrophy, it's actually a genetic disease that is caused by the absence of an enzyme in the peroxisms. Then the mitochondria. The mitochondria actually are going to be very important in energy production. We actually say that the mitochondria are the powerhouse of the cell, right? They are responsible for converting most of the food we eat from food energy to actually a form of energy that the cell is able to use. That form of energy is ATP, adenosine triphosphate ATP. That adenosine triphosphate is what fuels the chemical reactions that are happening in the body. For instance, I would not be able to run if I don't have enough ATP. I would not be able to do all the speaking I'm doing if I don't have enough ATP. I would not be able to think if I don't have enough ATP. So the mitochondria are responsible for the production of ATP. Now, I want to spend some time a little bit on some aspect of the mitochondrion. Notice that the mitochondrion as organelle is made up of two membranes. An outer membrane right here and an inner membrane that is folded into a very complex structure with the infoldings that we call cristae, right? The infoldings are called cristae. This infoldings is where the machinery responsible for harvesting most of the ATP that we use in our system are located, right? This is where the machinery responsible for harvesting ATP is located. And that machinery that is located in the inner membrane is nothing but the electron transport chain. Electron transport chain. That electron transport chain is responsible for the production of the massive amount of ATP our cells produce, right? The last thing I would like to mention about mitochondria is that when you consider all the cells in your body, beside the nucleus, the mitochondrion is the only other organelle that contains DNA as genetic information, right? So we have mitochondrial DNA. Now, let me tell you this. Your mitochondrial DNA in your body, this mitochondrial DNA comes from your mom. Your mitochondria, all your mitochondria, you inherit them from your mom. Your dad does not contribute when it comes to mitochondria, so that tracking the DNA that is in your mitochondria, you can tell who is the mother of an individual, right? But that's a parenthesis I wanted to mention. Another thing is that mitochondria are thought in the past to have their own life, right? We talk about the theory, the endosymbiotic theory. In other words, mitochondria used to have a life on their own, and then they got engulfed by a larger cell, and over time they became very interdependent. And at that point, in terms of energy, the rest of the cell depend depend very much on mitochondria. Now, in addition to the DNA that you find in mitochondria, you also have ribosomes. And here is the funny segment. These ribosomes that are found in mitochondria are very reminiscent of the ribosomes that are found in bacteria, right? That are not the same ribosomes as the one that are found in our own cells. So, when we talk about endosymbiotic theory, there is a real thought behind that. It looks like these cells used to live a life of their own. This slide is more of a summary slide. It's just to actually give you a way to summarize all the different organelles. You can think of the organelles: endoplasmic reticulum. What is the structure of the endoplasmic reticulum? It's a membrane network, right? Rough endoplasmic reticulum. It has ribosomes. The smooth endoplasmic reticulum does not have ribosomes, right? But now, on the other column, what is the function of the endoplasmic reticulum? It's a site of protein synthesis when ribosomes are present, right? And also lipid synthesis. Not added here, I should mention carbohydrate. Oops. So carbohydrate synthesis. I should add that. Okay? So please do the same thing for the rest of the organelles. Now let's take a look at the plasma membrane. The plasma membrane in humans are going to be made up of two membranes. You have one phospholipid layer here and another phospholipid layer here. And a phospholipid layer is characterized by the fact that it is made up of phospholipids, and these phospholipids have one head that is hydrophilic, meaning that it is water-loving, while we have a hydrophobic tail, meaning that it doesn't like water. When you put a lot of these molecules together in a water-based environment, that is the cell, well, needless to say, they are going to behave in such a way that the hydrophilic heads are going to be toward a water-based environment here, right? In and out. And the hydrophobic tails are going to also look toward each other so that the non-water-loving parts are going to stay away from water as much as possible, while the water-loving parts are going to get close to water, right? And this is how this phospholipid bilayer comes to exist. Now, in addition to the phospholipids, in human membranes, you are going to have cholesterol. That cholesterol plays a very important role in the fluidity of the plasma membrane. I'm not going to bug you with the details. Now, in addition to these lipids, the phospholipids and cholesterol, you're going to have some proteins that are embedded in the plasma membrane. These proteins that are embedded in the plasma membrane play a wide diversity of roles. Some of them are channel proteins, like here, right? Allowing chemical to go in and out. Some of them are proteins that allow the cell to sense what is going on outside and send the information inside. Some of them are coupled with enzymes. Some of them are proteins that allows the cell to attach to a specific matrix, right? Provided that they recognize that matrix, right? You have some sugars that are added to the proteins. These sugars that are added to the proteins are very important because they allow cells to tell each other apart. A skin cell knows that I'm in the midst of skin cell because of some surface markers they are able to recognize, right? Blood cell is able to recognize other blood cells because of the surface markers. These proteins and the carbohydrates that are attached are very allows the cells to tell each other apart, right? Whenever a protein has a complex sugar attached to it, we don't just simply call that protein anymore protein. We call it glycoprotein. So that's a protein to which a sugar is attached. By the same token, you have some lipids with sugars attached to them. These lipids with sugar attached to them are called glycolipids. Glyco means sugar. Lipid is lipid, obviously, right? Okay, so you have different components that make up the phospholipid bilayer, right? That phospholipid bilayer is fluid and is also characterized by the fact that it is it's a mosaic. What we mean by mosaic is that this this phospholipid bilayer is embedded with some proteins, and when you look at that uh, phospholipid bilayer with the protein, it is reminiscent of a mosaic, which is reminiscent of an artwork. Okay. Now, the proteins that are embedded in the plasma membrane, right? They contribute to cellular identity. I want you to remember that. Some of them are essential for transporting molecules in and out, right? Some of them are going to act as channels, some as transport proteins, some are essential for cell to cell communication as receptors, others are for cell to cell connection, right? So that the cells can connect to each other, okay? There are some genetic diseases where the the membrane proteins are not properly doing their job because they are not properly shaped. That's the case of what we call channelopathies. These channelopathies include, for instance, cystic fibrosis, right? In chapter 1, we talked about cystic fibrosis, where we say that the protein that is a membrane protein, in the case of cystic fibrosis, does not allow chloride ion to leave the inside of the cell to go outside. And because chloride ion is not leaving, water is not leaving either, right? Which causes the outside of the structure to have a thick mucus, right? Which causes the patient to be prone to diseases. Now, finally, but not least, I would like to address the cytoskeleton. The cytoskeleton plays a major role in many aspects, including the cell movement. For the cell movement, you are going to have microfilaments. These microfilaments are made up of protein units that are called actin molecules. These actin molecules are going to form a thread, and this thread end up forming very thin, complex filaments responsible for the movement, for instance, the crawling movement, right? When you consider your white blood cells that are going after harmful bacteria, right? They need to engulf them. They are able to crawl. The white blood cells, they are able to crawl because of the action of the microfilaments. Now, how about the microtubules? The microtubules are filaments that are relatively thick and rigid. They are made up of building blocks that are called tubulins. The tubulins occur in two kinds, the alpha and the beta tubulins. I'm not going to bug you with the details, but I want you to remember that the tubulins are responsible for the development of microtubules. And microtubules play a very important role in cell division because the microtubules are responsible for distributing the genetic information equally during cell division, right? They play a very important role in cytoplasmic streaming. In other words, when we consider a cell at a particular instant called instant T, at that particular instant, the nucleus is at a given location, the Golgi apparatus is at a given location, the endoplasmic reticulum is at a given location. This is made possible because of the microtubules. Also, the microtubules actually are able to stand as railroad tracks for transport vesicles. For instance, how does a transport vesicle that is carrying a cargo, a protein, toward the outside of the cell knows that the outside is this way, right? For the vesicle to move in the very specific direction it is supposed to move, well, it does not move there randomly. The microtubules are actually standing as tracks, and these vesicles are moving along that track, right? Finally, I want to talk about the intermediate filaments. The intermediate filaments are the least well characterized filaments of the three micro cytoskeletal filaments. However, the intermediate filaments play a very important role, for instance, in strengthening the membranes, providing a mechanical resistance to environmental influence. Finally, let's talk about the cilia and the flagella. The cilia exist on the surface of some epithelia, such as, for instance, the trachea. Along the trachea, you have epithelial cells that are columnar and that have the cilia. The cilia vibrate. They vibrate so that they can push the mucus in a very specific direction, right? These cilia are made up of microtubules, and these microtubules, as they are fueled by ATP, are going to vibrate with a whip-like motion, right? Finally, but not least, you have here sperm cells, and these sperm cells have a head and a tail. That tail is actually made up of microtubules. These microtubules, when they are filled with ATP, they're going to be engaged in a whip-like motion and propel this and propel the sperm cells toward the egg so that they can go and fertilize the egg. So this is where we are going to stop the first part of this chapter. Thank you for your attention.